Howdy. I have to do a project for my biology class. I decided to make a video about enzymes. That's this video. First upload in 4 months, and the video is enzymes. Yay. Speaking of which, I might as well start. So, first of all, what exactly is an enzyme? That's a good question. To understand this video, you need to know what an enzyme is. Simply put, an enzyme is any number of biological macromolecules, proteins specifically, that are used to catalyze chemical reactions. By any number, I mean there are many tens of thousands of them. Around 75,000, if my memory serves me correct. Okay, I'll give you a moment to digest that. A lot of big words thrown at you in a short period of time. I imagine many of you have heard of proteins. It's one of the things that are on the food labels. You may have heard it helps you build muscles, etc. Proteins are a class of molecules that are made up of chains of amino acids. They come in a few different classes and are put into those few different classes depending on the structure of the protein. Catalyzation, or catalysis, is, in essence, the process of lowering the amount of energy required to perform chemical reactions. A reaction's activation energy, if you will. So, I'll restate my former claim. Enzymes are proteins that catalyze chemical reactions. They speed up chemical reactions. That should be easy to comprehend. The way I learned enzyme structure was the so-called lock and key model. It compares the enzyme to a lock and the substrate/reactant, two words that mean the exact same thing, to a key, which at the surface level makes sense as different substrates can only bind to certain enzymes. An important thing to know about all proteins is that structure determines function. However, it's not the enzyme aspect of the analogy that bothers me. Locks and keys are quite easy to break. Go watch a lock-picking lawyer video if you need evidence. After you finish this video, of course, you are going to watch to the end of this video, right? Good. Sorry, I got sidetracked. In short, enzymes are unlocked by substrates, and each enzyme only reacts with a specific complementarily shaped substrate. The substrate collides with the enzyme at the active site, an area where the shapes of the enzymes and substrate allow them to bind to one another. However, the active site is only on the enzymes, not the substrate. I'm telling you this because I was told this several times, so apparently it's important information, and who am I to disagree with a Midwestern suburban high school science teacher? Catalyzation can be described in 4 concise steps. The substrate enters the active site. The substrate and enzyme bind together to form an enzyme-substrate complex. The enzyme alters the substrate, and it becomes an enzyme-product complex. Finally, the products leave the active site, and the enzyme reverts to its original pre-complex form. The amount of time taken to describe what the enzyme and substrate do is 1/4 of the words I use to describe how they do it. Enzymes are fickle beasts, however. Recall that the function of proteins is determined by their structure. That is basic, fundamental biology. Enzymes, being proteins themselves, only work when their structure, their form, are intact. There are a few factors that can affect how efficiently enzymes work. 3 big ones are temperature, pH level, and the concentration of enzymes and substrates. Those should all make sense. Temperature is obvious. There were a whole 2 pages in our note packet dedicated to what temperature does to enzymes. In those pages, we had to come up with a list of at least 3 things that stop working properly if they melt or change shape, as, a little comparison. My list was, and I quote, 1. Fork. 2. Coin. 3. Grandpa. I'll throw up a picture on the screen of the page. Enzymes, when they are heated to a high enough temperature, denature. Their molecular structure warps and malforms to such a degree that they can no longer catalyze reactions. When an enzyme is denatured, substrates cannot enter the active site. The enzyme amylase, for example, is used to break down starch into more basic sugars during eating. It is found in the mouth. so tends to like things toasty. My sources vary, but around 37 degrees Celsius toasty. That's 98 Fahrenheit for destiny manifestors such as myself. In layman's terms, they like it hot. However, if it gets too hot, they naturally denature. If you say light your mouth on fire, you won't be able to digest potatoes. Shame. The effect of pH level on enzymes should also make some semblance of sense. As with anything organic, bad stuff can and will happen if the environment is too acidic or basic, specifically, once again, denaturing. However, the favorable conditions for each enzyme can vary quite a bit. This seems like a good place to interject and talk about buffers for a moment. Most of the human body has a nice neutral pH of 7, and we like it like that. If it were different, most biological processes of ours would not work. Amylase, which I will again use as an example, has an optimal pH of 6 to 7, and as such should not be surprising that the human mouth has a resting pH of 6 to 7. If a mouth were to become significantly acidic or basic, the amylase in that mouth would cease to function. There is, of course, always the risk of such a thing happening, but the body has a system in place to counteract it. Buffers, as they are called, are aptly named. Buffer solutions are solutions, homogeneous mixtures, that are meant to resist changes in pH levels throughout the body. To explain in depth would require explaining exactly what acids and bases are and how to define them, so I'll try to keep things pretty surface level. If you want me to talk about acids and bases, tell me in the comments or on the Discord server. Essentially, acids are solutions with more free-floating hydrogen ions, written as H+, whereas bases are solutions with more free-floating hydroxide ions, written as OH-. I don't know if you can hear that in the background, but like the tornado sirens just started playing. It's, it's a Saturday. They do that. They test stuff here. Make sure it works properly. Sorry if you can hear that. If you can't hear that, just disregard what I'm saying entirely. Buffer solutions work by absorbing excess of these or releasing them when there is not enough, as hydrogen and hydroxide ions react to form water, which has a neutral pH of 7. An example is the bicarbonate buffer system. It's used to maintain a standard pH in the blood and works by reacting water with carbon dioxide to form carbonic acid, which deforms into a hydrogen ion and a bicarbonate ion, HCO3-. The process also works in reverse, where if a bicarbonate ion and a hydrogen ion react, they can form carbonic acid, which can then turn into water and carbon dioxide. Put simply, the bicarbonate buffer system counteracts changes in blood pH by forming carbonic acid and letting that carbonic acid separate into its constituent parts. Buffers in general counteract changes in pH by utilizing chemical reactions to absorb or release hydrogen or hydroxide ions. Now that we've touched on buffers, I feel comfortable talking about pH and enzymes. Most enzymes prefer a pH of 7 because that is what most of the human body is. Not all, however, there are naturally some exceptions. The enzyme trypsin, which can be found in the small intestine, is used to break down proteins. It prefers a slightly basic environment, being most active at a pH of 8. Trypsin would not work well in, say, the mouth, which is slightly acidic and would probably be completely broken in highly acidic conditions, such as the stomach. However, some enzymes can survive the hydrochloric conditions of the stomach. Pepsin specifically prefers a pH of around 2, conveniently close to the pH of gastric acid, where it can be found. The word pepsin may remind you of Pepto-Bismol, and that was intentional. Both words derive from the Greek peptine, which means to digest, and that's exactly what pepsin does. It breaks down proteins for easier digestion in the stomach. Pepsin apparently likes to be on the verge of complete and utter annihilation. I respect it. However, if someone were to say, eat some lye, which has a pH around, I want to say 13, It could possibly neutralize someone's stomach, or perhaps even make the stomach slightly basic. If that were to happen, that person's pepsin would obviously not work. Neither would the rest of the person. Obviously, the concentration of enzymes and substrates is also important. The more of any specific enzyme there is, the more reactions can be carried out at any one time. With substrates, it's a bit different. Yes, the more substrates there are, the more reactions that can happen, but without increasing the number of enzymes, eventually the rate of reaction will plateau. The specific limiting factor I'm not going to go into as much detail with, as the information should be obvious. Enzymes and substrates are needed for reactions, so more of them means more reactions, and vice versa. There is one last enzyme limitation I would like to talk about. Or two, I guess. Inhibitors. Enzyme inhibitors are molecules that bind to enzymes to prevent catalyzation. There are two types, competitive and non-competitive. Competitive inhibitors are molecules that bind to the active site of the enzymes, which prevents substrate from binding to the enzyme. These are typically reversible and are most commonly reversed by adding more substrates. Some examples are caffeine, aspirin, and many types of nerve gas. There are also non-competitive inhibitors, which do not bind to the active site. Instead, they bind to another part of the enzyme called the allosteric site. In doing so, they change the shape of the enzyme, which, whilst allowing the substrate to enter the active site, the substrate is nonetheless prevented from binding, which prevents catalysis. Non-competitive inhibitors are typically harder to reverse than competitive inhibitors, sadly. Some examples are these words I cannot for the life of me pronounce. Now, my class didn't touch on this, but in doing my research for this video, I learned that competitive inhibitors can actually be allosteric, meaning they bind to the allosteric site. To be a competitive inhibitor, the inhibitor only has to prevent substrates from entering the active site. The more you know. That brings me to the end of our little enzymatic journey. I hope you liked it. I, believe it or not, actually enjoyed making it. Subscribe, comment, do whatever the heck it is YouTube people do. Look in the descriptions if you want to join the Discord server, I guess. That's all I have. Enzymes are proteins that speed up chemical reactions. That is probably the most important information in this entire video. Hold on to that. Well, have a good one. Bye.